Hello everyone. Today we will discuss about the part 7 of the important notes for the biochemistry so that you can easily able to answer the one-liners and the MCQs in your exam. First of all, what is link reaction? Link reaction, that means conversion of the pyruvic acid into acetyl-CoA inside the mitochondrial matrix. So link reaction, that means the conversion of the pyruvate to acetyl-CoA, and remember this link reaction is actually a link between the glycolysis and the TCA cycle, or we can say tricarboxylic acid cycle. Now this link reaction occurs in the mitochondrial matrix, and the link reaction step is oxidative decarboxylation, where it basically requires vitamin B1. So as you can see in this figure, this pyruvate actually enters inside the mitochondria, and the pyruvate is then converted into the acetyl-CoA with the help of this link reaction. Now this link reaction occurs in the fed state. Link reaction is activated by the insulin. Now pyruvate enters the mitochondria via the pyruvate proton symport present in the inner mitochondrial membrane. Link reaction enzyme is the PDH complex or pyruvate dehydrogenase complex. Now pyruvate dehydrogenase complex is a multi-enzyme which actually contains three components, that is the enzyme 1 PDH or pyruvate carboxylase, enzyme 2 that is the dihydrolipoyl transacetylase, and the enzyme 3 that is the dihydrolipoyl dehydrogenase. Now the pyruvate dehydrogenase complex and the tricarboxylic acid cycle both requires 5 coenzymes, that is vitamin B1, B2, B3, B5 and the lipoic acid. Deficiency of these actually leads to the decreased energy production and the central nervous system problems, as the brain cells over here depends upon this aerobic reaction. Allosteric inhibition of the pyruvate dehydrogenase occurs by acetyl-CoA, NADH and the ATP. Pyruvate dehydrogenase is active in dephosphorylated state, and dephosphorylation is done by insulin. Arsenate inhibits the enzymes requiring lipoic acid as coenzyme, such as pyruvate dehydrogenase complex. Deficiency of the pyruvate dehydrogenase complex is the most common biochemical cause of the congenital lactic acidosis. Beriberi causes lactic acidosis. Link reaction is irreversible. Tricarboxylic acid cycle is both anabolic and catabolic, that means it is a amphibolic pathway. Tricarboxylic acid cycle is actually a vital cycle and it has no hormonal control. TCA cycle cannot occur in the anaerobic conditions and is strictly aerobic. TCA cycle occurs both in the fed as well as in the fasting state. TCA cycle neither activated by insulin nor glycogen. TCA cycle occurs in all the cells of the body where the mitochondria is present. So we can say this particular reaction actually occurs in the mitochondrial matrix. That's why this particular cycle actually requires those cells, or we can say this occurs in those cells only where there is mitochondria. So remember, it occurs in the mitochondrial matrix. TCA known as tricarboxylic acid cycle because over here the citrate and the isocitrate are tricarboxylic acids. Krebs cycle name is by name of the scientist, hence Adolf Krebs. So this tricarboxylic acid cycle is also known as the Krebs cycle. TCA is also called a citric acid cycle, as first compound formed is the citrate, which is a prochiral molecule. Prochiral molecules are those which can be converted from achiral to chiral in a single step. Citrate inhibits glycolysis and activates fatty acid synthesis. Now in the Krebs cycle, 4 steps are involved in oxidation reduction, and the enzyme involved over here are the isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase and malate dehydrogenase. Over here, 2 steps involved in the oxidative decarboxylation, and over here the enzyme for this particular oxidative decarboxylation is the isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. Oxaloacetate is regarded as the first substrate and carrier of the TCA and not acetyl-CoA. Oxaloacetate has a catalytic role in the tricarboxylic acid cycle. Acetyl-CoA is not the intermediate of the TCA cycle and is never glucogenic. Aconitase always acts on the part of the citrate derived from the oxaloacetate. Fluoroacetate is non-competitive inhibitor of the aconitase. Fluorocitrate is competitive inhibitor of the aconitase. Aconitase or the aconitate hydratase belongs to the lyase category and is exceptionally an iron sulfur protein. Alpha-ketoglutarate dehydrogenase is a multi-enzyme complex similar to the PDH complex or the pyruvate dehydrogenase complex and requires same 5 coenzymes, that is the thiamine pyrophosphate, lipoic acid, FAD, coenzyme A and the NAD. Alpha-ketoglutarate dehydrogenase is not regulated by phosphorylation or dephosphorylation. Enzymes catalyzing dehydrogenation without decarboxylation do not require metallic ions for their activity like magnesium or manganese. Substrate level phosphorylation step. Now over here the SLP step, there is conversion of the succinyl-CoA to succinate by succinyl-CoA synthetase or the succinate thiokinase. Now over here there is production of both ATP and GTP, but ATP is the predominant one. GTP is produced in the liver and the kidney during the starvation process for PEP-CK enzyme of gluconeogenesis. There is only one substrate level phosphorylation step in the TCA cycle. All enzyme of the TCA cycle lies in the mitochondrial matrix except for the succinate dehydrogenase, which basically locates in the inner mitochondrial membrane. Succinate dehydrogenase is a flavoprotein enzyme in which the FAD acts as hydrogen acceptor and is inhibited by the malonate. Succinate dehydrogenase is involved in the TCA cycle as well as the ETC, or we can say the electron transport chain. Fumarate hydratase or the fumarase and aconitase are lyases. 1 acetyl-CoA gives 10 ATPs in the TCA cycle, where the 9 ATPs are by the oxidative phosphorylation, or we can say ETC, and 1 by the substrate level phosphorylation step. Anaplerotic reactions are those reactions which replenish the TCA intermediates. Most important anaplerotic reaction is the pyruvate to oxaloacetate conversion by the pyruvate carboxylase enzyme. 2 carbon dioxide in the TCA are derived from the carbons of the oxaloacetate. Irreversible steps of the TCA cycle are the, remember, citrate synthase and the alpha-ketoglutarate dehydrogenase. There are 3 rate limiting steps in the TCA cycle depending on the various complex conditions in the body, and they are citrate synthase, isocitrate synthase and the alpha-ketoglutarate dehydrogenase. So today up till here, we will discuss about the further important notes for the biochemistry in our next video. Thank you so much.